Hi, Rich.
Hey, sorry I'm late. I had just finished another meeting.
You're actually the first.
Great. So I'll get going now that we're on streaming. And Tim, you said you were able to keep an eye on that, so thanks very much. Cool. So welcome everyone to today's Earth2Earth seminar. Thank you very much for joining us today on the 11th of February. As a reminder, the mission of Earth2Earth is to engage the UK geoscience community with inspiring science from internationally leading researchers, promoting inclusivity and diversity, and supporting early career researchers and geoscience's contribution to addressing global environmental challenges. It's open to all UK institutions, so please, if you're interested in helping organise, do get in touch. Just for kind of technical side of today's seminar, if you've got questions, you can put them in the Q&A at any time during the meeting. We'll come to them at the end. We'll pitch them to Barbara then. So it's my great pleasure to introduce today's speaker. We're fortunate today to be joined by Professor Barbara Sherwood Lollar. Barbara is a Professor of Earth Science at the University of Toronto, where she holds a Canada Research Chair in the Isotopes of the Earth and Environment and the Norman Keeble Chair. Barbara's work has involved a really wide-ranging investigation of stable isotope geochemistry as applied to high and low temperature planetary systems, the remediation of drinking water supplies, and the study of deep subsurface microbiology. Barbara's been president of the Geochemical Society and in 2019 was named a fellow of AGU. And today, Barbara will be introducing us to the hidden hydrogeosphere and the search for life. So thank you again, Barbara, for joining us. Please, over to you.
Thank you so much. I really appreciate the invitation. Exactly a year ago, I was with you in the UK. It was my last trip before lockdown. Had been actually a trip over to visit with colleagues and students and postdocs at Oxford. So I wish I could be with you in person, and hopefully we all come through this and someday we meet again. But in the meantime, of course, we take advantage of this technology, which does allow us to reach out wider and deeper. And so I really appreciate the opportunity and hope that we can make this work. I'm going to jump to share screen at this point. And please just shout out if at any point there's any difficulty with the presentation. So at the moment, I hope you're actually seeing my screen. Is that correct? Yes?
No, at the moment
Still
we
not.
can't.
We'll try again. That's interesting. Try the share screen function again. Let's see where we're had it working before. According to this, it says I'm screen sharing.
Great. Yeah,
we can see my screen now. Okay, great.
Yeah.
Excellent. Then let's go to that. All right. Okay. Perfect. Terrific. So as Oliver mentioned, I'll be speaking about a range of the work that we do in the hydrogeosphere. I'm an isotope geochemist with applications to understanding the study of groundwater and fluids. And this particular talk will focus more on the deeper aspects of the work that we do, with its crossover links to subsurface microbial life and indeed to astrobiology and planetary sciences. I'm going to kick off with this study that was done by some colleagues at the University of British Columbia in Canada a couple of years ago. Now, they were focused on something very different. They're interested in understanding modern groundwater. And so they did this interesting study using tritium to try to understand the global distribution of what they called modern water, that is water less than 50 to 100 years old, and down to about 2 kilometers in the crust. And it was a study that got a lot of attention at the time. It's very nice and elegantly done. And that was largely because what they were able to show is illustrated quite nicely in this graphic. Can you see my mouse as I do this or no? You can. Terrific. They were able to show that their estimate of the total inventory of modern groundwater, that is groundwater less than 50 to 100 years old, is this giant bubble here. And that setting aside the oceans, which are of course the largest water inventory, if instead one looks at everything else, that modern groundwater was by far the largest proportion of the hydrogeosphere compared to the atmosphere, vegetation, surface water, etc. But what I found fascinating about this study that didn't really come out at all, but to me, the part that really engaged me is that if you took a look at this estimate of modern groundwater, what you found is in fact expressed in the other half of this image that they showed. And that is, if you take all of this modern groundwater, less than 100 years old, and compared it to total groundwater estimates in the subsurface, it corresponds to this tiny drop in what is otherwise the huge amount of groundwater that's much, much more than 100 years old. And yet this didn't get talked about. What was fascinating here is that modern groundwater really only accounts for a couple percent of total groundwater estimates to 2 kilometers. And the unappreciated corollary is that the vast majority, 97 to 99% of total estimated groundwater, is older than 100 years, is not actively participating in the hydrological cycle on short timescales. And in fact, we know very, very little about it, including how old it is, what the long timescales of the hydrological cycle are, and indeed what the subsurface inventories are. So a lot of the work that we've been doing is really within that context, the context of trying to understand this, what we call hidden hydrogeosphere. And I think we first used that term in a paper by Oliver Warr, who's a research associate in our lab and a UK resident, a graduate of Oxford and Manchester, who's now working with us at the University of Toronto. And it really, these are some of the large order questions that we're struggling with in our research. Understanding the rest of that groundwater, the stuff that's more than 100 years old, understanding its volume, not just to 2 kilometers, but to 5, 10, and even deeper levels in the crust. Understanding the age distributions. Where do we see young groundwater? Where do we see older groundwater? And what are the controls of depth, lithology, and structure? And indeed, I'll talk a lot about this today in the context of one of our sites, the Kidd Creek setting in Canadian Shield. What are the oldest fluid components that are macroscopically stored in the crust? So looking beyond just fluid inclusions. And indeed, this hidden hydrogeosphere has a driver in terms of the hidden biosphere, the deep subsurface biosphere, because it's only by understanding these fluids that we can really put some important constraints on our understanding of subsurface microbiology. And so some of the other questions that we struggle with here in context of the deep biosphere are expressed here today. I won't be focusing as much on the biological aspect, but nonetheless, the whole driver behind some of these water-related questions are in the context of understanding water-rock reactions that sustain life, the production of reduced gases like hydrogen and methane deep in the earth, and the implications all of this have for habitability and subsurface life. The other point that I should make is, of course, a lot of similar research goes on in the marine lithosphere. What you'll see today is our focus is primarily on the terrestrial, the continental lithosphere for the deep hydrosphere and biosphere, and indeed specifically on the Precambrian crust. And there's a really good reason for that. If you take a look at the distribution of Precambrian rocks, you'll see that they account for more than 70% of the continental lithosphere by surface area. By volume, it's even larger, estimated at 88 to 90% of the continental lithosphere. So if we really want to understand continental subsurface life, then the Precambrian crusts are indeed an excellent place to look. These are tectonically quiescent, of course, in the present day and represent some of the oldest rocks on Earth. We access these primarily by using subsurface exploration, either boreholes, subsurface research labs built for scientific purposes, or indeed active mines that allow us to go down 2 and 3 kilometers into the crust to take samples. And the work that you'll see today is a composite of data that's been derived from our work over the last 30 years in both Canada, South Africa, Fennoscandia, and most recently in Brazil. Although I won't talk about the Brazilian data yet, it's still under development, but we've recently been doing fieldwork there. What do we know then? To give you a quick overview of all of that work over 30 years, what do we actually know at this point in time about the distribution of water, energy, and life in the world's oldest rocks at depth? What's fascinating about this topic is that this is something that's really run underneath the scientific radar, but it's a phenomenon that's been well known to anybody who's explored these rocks over time. The earliest records of this phenomenon are found in Canada, at least, in the Annals of the Geological Survey, or more than 100 more years ago. They noted that gas and water was found in vugs or cavities in these deep rocks under pressure. And indeed, if you talk to miners throughout the world doing work in the many economic mines throughout the crystalline rocks of the Precambrian shields, they know about this. This is a naturally occurring phenomenon. It's not related to the mine itself. These are discharges of saline water naturally occurring in these rocks. And again, known to the mining community, but very much running underneath the radar of the scientific community until about the 1980s, when a lot of these environments began to be investigated not only for economic exploitation, but for radioactive waste disposal, actually. So what do we know then about these fluids now that we have actually begun to put them underneath the scientific microscope? Again, they are highly saline, typically calcium, sodium, chlorine dominated, typically 50 to 180 or even 200 milligrams per liter salinity. So many times the salinity of seawater. Depending on where you are, different temperature gradients, water flow rates can range. Sometimes when these fractures are first opened up because of the excavations, you can get flow rates on the order of tens to hundreds of liters per minute. It can be quite a explosive phenomenon. But typically those high flow rates do die down, and over long time scales, we see flow rates more on the order of liters of water per minute and liters of gas per minute coming out of these boreholes, ranging from very gentle bubbling, such as you see here in these two images, to something more like this. This one video that I'll show you shows the flowing water on the order of about a liter of water per minute and about a couple of hundred liters of gas per minute. This is taken from 2.4 kilometers below surface on the Precambrian shield. And so you can see from that then the visible manifestations. We put it through that plexiglass tube so we can see the gushering as the exhaled gases degas from the water as these things flow when released. And that particular system has actually been discharging at about that rate of flow since we first discovered it back in 2007. In terms of the geochemistry of these fluids, in addition to the aqueous geochemistries, which I've just given you a small glimpse into, we also see high concentrations of dissolved reduced gases in these systems. In particular here, the picture that I've just shown you from Kidd Creek on the Canadian Shield at 2.4 kilometers below surface, I'm comparing here to something you may be more familiar with, which is the Lost City hydrothermal vents on the ocean floor. And as you can see here, if we look at dissolved hydrogen associated with Kidd Creek and with Lost City, you can see they're very comparable. Kidd Creek also has high concentrations of methane, ethane, propane, and butane. But for the purposes of what I'll focus on primarily today, I really am going to focus primarily on the hydrogen. And so this just gives you a small taste of exactly how hydrogen-rich these systems are. On a global scale, this is characteristic of these saline fluids. Whether you are in Finland, South Africa, or Canada, we see that, like the hydrothermal vents, these are just some of the most hydrogen-rich environments on the planet, with concentrations of hydrogen on the order of 5 to 15 millimoles per liter. In the dissolved gas phase, that means you can have concentrations as high as 40 to 50% by volume in the gas phase. And as you can imagine then, with all of that hydrogen hanging around, these are very attractive places for microbiology and have therefore been the subject of significant study in terms of understanding deep subsurface microbiology. Some of those key studies are shown on the lower left. There are many, many more by groups all around the world who have been investigating these hydrogen-rich, energy-rich deep subsurface fluids. And down to about 1.5 kilometers, there are now literally dozens of studies of those microbial communities. But it is important to point out, as you go deeper than about 1.5 kilometers, as you get into some of these waters that are 2 and 3 kilometers deep, this is where we see some of the most saline end members, some of the oldest groundwaters, some of the most hydrogen-rich systems. To date, those have been significantly less investigated in terms of their microbiology. There's a handful of studies to date, lots of room for more scientific investigation amongst the young folks who are out there and looking for interesting targets. What we do know is that those deepest, most ancient and saline fluids are characterized by microbial communities, if they are present, that are characterized by low biomass, low biodiversity, and although there are a variety of metabolisms that are manifested, we typically see a dominance of hydrogen-utilizing sulfate-reducing organisms. That's a very typical organism that one finds if one does find life in these systems, and you don't find it everywhere. And so The geochemical context, the hydrogeological context that I'm putting forward has really been a driver for us in terms of our search strategies for subsurface life, where to go looking for it, where to understand the nature of life in the deep continental lithosphere. And essentially, we have expressed this kind of in the same context, a little bit tongue-in-cheek, but the same context that we use for investigating Mars, this idea of follow the water. Because in fact, it's a very powerful scientific strategy. And interestingly, though, one that's still underutilized, even within the subsurface microbiology community. And that's partly expressed here in the context of there's really two components to that idea of follow the water. We definitely immediately, I think, have a natural reaction to the first bullet, the idea that life needs water to survive. But in fact, there's a corollary to that that we sometimes think about less than we ought to, which is that habitability and the presence of subsurface life requires more than just the presence of water. It actually requires the ability for that water to flow and mix on some kind of a geologic timescale. In other words, it's deeply dependent on the concept of hydraulic conductivity. In near surface systems, whether that's on the continents or in the ocean floor, advection and diffusion really controls the flow of groundwater, largely because water is largely found in the pore spaces in between the pore grains. As you go deeper into these kinds of crystalline rock, though, you really change the nature of the hydrogeologic setting, and you enter into a very different system in which hydraulic conductivity is actually controlled not so much by the porosity between the grains, but instead by the fact that the water is predominantly trapped within fracture networks in these crystalline rocks. And in fact, as you go deeper into these systems, as we do, you enter into systems like this, in which those fractures are actually discontinuous and sparse. It's essentially a system described by hydrogeologic discontinuity. And as you can imagine then, if the distribution of water in these systems is patchy and irregular, then the distribution of life will be patchy and irregular as well. And these fractures provide us, in fact, with, in a way, time capsules, a variety of different systems that we can investigate because those fractures represent different degrees and timing of either isolation or connection to surface. And so it's important to emphasize as well that in this kind of a system that represents water and life in isolation in space and time, that these fractures are not static. They do, even in the absence of plate tectonics or recent tectonic activity, these fracture systems do open and close due to changes in stress and the precipitation of fracture minerals. So this provides a mechanism by which these networks, in fact, capture fluids and gas of different provenance and age. And as we've investigated them through the years, we have found, in fact, you can identify systems that have residence times on the order of not just hundreds of years to thousands of years, but typically as you go deeper into these systems, this is where you begin to encounter groundwaters that have residence times on the order of millions and even billions of years. So the fracture networks present a temporal and spatial variations in the supply lines for life. And we've seen this, for instance, in terms of our understanding of some of the deep communities identified in our work in the South African gold mines with colleagues there, where we found that understanding the nature of the water and being to understand that those waters have residence times of hundreds to tens of millions of years has been part of understanding the degree to which the microbial communities associated with that waters have also been cut off from the photosphere on very long geologic timescales. So the noble gases then have been a key tool in order to allow us to provide constraints on the water and then by context on the life in these systems, either providing us with evidence that we're looking at a system that has had recent connection to surface, or alternately providing us with the lines of evidence that show us the timescales of hydrogeologic isolation. This group, I'm sure, needs no introduction to the noble gases and why they're such a powerful tool. They're, of course, the inert, the conservative part of the periodic table, and allow us to identify and quantify the origin of floating gases based on a very well-characterized end members. The helium, neon, argon, krypton, and xenon can be derived from the mantle, from the atmosphere, or produced by radiogenic reactions in the crust. And it's really the latter category, the radiogenic noble gases, that allow us to put these residence times estimates on these fluids. There's a very well-known technique. I won't go through it in detail for today, but I did provide a nice review paper from our collaborator, Chris Ballantyne and Pete Berard. They did a very nice review paper here that gives you the background behind how all of these measurements of the radiogenic noble gas and the ingrowth of those gases into fluids and gases in the crust provides us with a means of estimating residence time. What it all adds up to then is an ability to put this kind of temporal constraint on our understanding of deep water and deep life. And indeed, as I've mentioned, we have a variety of studies through the years. We're interested in these waters regardless of their age, and they do range from thousands of years to millions of years. But what I'll focus on today is in particular the kind of oldest end member sites, and in particular a site called Kidd Creek in the Canadian Shield, which I've mentioned already, because indeed it was there that a few years back we were actually able to really push back our understanding of just how old waters can be. But we did that not in the context of going out to try to break a record. We were doing it in the context of this overall study, which I mentioned to you, which uses this as a very important tool for understanding the nature of water and the nature of life in the crust writ large, whether it is in thousands of years or millions of years, or in this particular case, this fascinating setting where we actually found residence times in the order of billions of years. Kidd Creek then is really where I'll focus for the rest of today's talk. And for that, I'll give you just a quick overview of where it is. It's located about 8 hours drive north of Toronto in the 2.7 billion year old rocks, the Archean rocks of the Canadian Shield. It's fascinating for many reasons, one of them being it's one of the deepest base metal mines in the world, with active workings down to 3 kilometers and drillings even deeper than that. It's also one of the most well-preserved sections of the 2.7 billion year old crust. Relatively little metamorphosed, only to green schist facies, relatively little deformed. You can actually still see the pillow lava structures in the rock that show you that this was deposited on the ocean floor 2.7 billion years ago. But what has happened is that ocean floor has been entirely inverted, and what was originally laid down is a horizontal deposit along the ocean floor, as you see, has now been completely inverted. And so this is the reason why they have to drill and excavate to depth as they follow those down. We had the opportunity at that time to investigate a series of boreholes. In fact, the one for which I showed you the video earlier is just one of a series of about 5 or 6 boreholes that we've now been monitoring over almost 15 years at an observatory set aside for science within the operating mine, 2.4 kilometers below surface. In this study, by working with Chris Ballantyne and Greg Holland back in 2003, we monitored those fluids for their noble gas contents over about 12 to 15 months. And to cut a long story short, the key finding was this: when we took a look at the 4 independent estimates of residence time from the radiogenic helium, neon, argon, and xenon, all of those estimates provided us with mean residence times of more than 1 billion years in age. This was some of the oldest residence times ever derived for these kinds of deep subsurface fluids. It's since been confirmed not only at Kidd Creek, but in other systems like this around the world. But it really pushed back our understanding of just how old fluids can be. If you take a look at that paper and ones to follow, you'll see that the noble gases were just one of multiple lines of evidence supporting the antiquity of these fluids. Additional lines of evidence included Archean, neon, and xenon isotopic signatures, noble gases from some of the other means by which they can be derived in these systems as well. And indeed, simply looking at the O18 and deuterium signatures for these fluids, they also showed us some of the most elevated levels over the global meteoric water line that had ever been identified before. So multiple lines of evidence supporting that antiquity, and literally allowing us to understand something about the storage of ancient remnants and components of fluid and gases in the crust. Geologists are, of course, used to this concept from the point of view of fluid inclusions. We've long known that remnants of ancient fluid and gases can be trapped in a microscopic means within fluid inclusions in the rock. But this was really the first time we'd understood that in some special places, particularly where there's been a long-term preservation, that there's an opportunity for some of these ancient noble gases to also be preserved on a macroscopic scale. and to actually be present in fluids that are discharging at rates that allow us to fill up liters worth of sample. So it's actually changed the nature of the samples that we can utilize and investigate to understand something about fluids on the ancient Earth. That said, what I'll focus on primarily today is in the context of the microbiology side of this, the life search and the implications of that for planetary sciences and astrobiology. So I'm going to move back now to focus less on the details of the noble gases, now that you understand the context in which we use them, but to go back to this point that we made earlier, that these fluids are present in Precambrian and Archean rock throughout the planet, that this constitutes 70% of the continental lithosphere by surface area, and that these fluids are not only saline due to the very long-term water-rock reaction, but contain high concentrations of hydrogen. This in itself drove a major change in our understanding of the habitability of our planet. Up until this point, when one is looking at hydrogen-driven deep life, the focus had primarily been on marine systems and the hydrothermal vents. This really changed our focus, and for the first time we recognized that these kinds of hydrogen-driven microbial metabolisms might be enabled within the deep continental systems as well on a global scale. And in fact, in that paper from 2014, we took a look at it from the point of view of doing exactly that, not understanding these sites just on a site-by-site case, but actually calculating for the first time what this might actually mean on a global scale. And the calculations in that 2014 paper derived on understanding the water rock reactions producing hydrogen in these systems. There's a wide variety of these kinds of water rock reactions. Some of the most obvious are, in fact, a very well-known process to geochemists, the process of serpentinization. Going back to work of Barnes in the 1970s, we understand that in ultramafic and some mafic rocks, in fact, when you have hydration of those rocks, that by these various reactions, hydrogen is a natural byproduct and can build up in these kinds of ultramafic systems. In addition, though, there's a second major category of hydrogen-producing reactions that we've seen, and these are found in these kinds of rocks even where you don't have ultramafic rocks. So these are common to all of these lithologies, and that is the process of radiolysis, by which uranium, thorium, and potassium in the crust and their decay, of course, produces alpha, beta, and gamma particles. And when those attack the water molecules, you get cleavage of the water molecules to produce hydrogen. And this is seen all around the world as well. And so depending on where you are in these rocks, in the Witwatersrand Basin, in particular where we were working, these were not particularly ultramafic rich systems. These were more felsic sites, and most of the hydrogen in those systems was coming from radiolysis. Whereas on, for instance, the Canadian Shield, we see both these famous greenstone belts of the Abitibi province, where we have a lot of ultramafics contributing to hydrogen production by serpentinization, but in the other lithologies, a second contribution from radiolytic reactions as well. There are other reactions that produce hydrogen in these systems, but those are the two big categories that account for hydrogen production in these systems. And what we did in that 2014 paper was really to take hydrogen data from these kinds of sites all over the world, from our own work and from others, and to bring them together in a global compilation of about 350 different samples and measurements. And that allowed us to actually do a first estimate of the global production of hydrogen from the combination of these water rock reactions for the continental lithosphere. By doing that, we were able to show that, in fact, by focusing just on the marine lithosphere, we've been missing about half of the global hydrogen production from these reactions. So just by focusing on the continents for a minute, we really were able to double our understanding of global production of hydrogen from these reactions. And again, that's critical in terms of our understanding of habitability, of where and how on this planet we can sustain subsurface life. One of the other issues that I'll focus on in today, of course, is that hydrogen alone just gives us information on the electron donor. I'd mentioned earlier that some of the deep subsurface communities are dominated by hydrogen-utilizing sulfate reducers. So as you can imagine, amongst the variety of other reactions and other questions that we're asking ourselves, one of the key questions was not just to understand the origin of hydrogen in these systems, but to understand the origin of sulfate as well. Because of course, any organism is going to need both to survive. This work was carried out by Long Lee, who was a postdoc in our lab at the time, but is now a tenured professor and CRC chair at the University of Alberta. And Long was very interested in taking a look at these rocks and fluids from the perspective of sulfur isotopes, as you've seen here. As we know, when we look at the sulfur isotope signatures within the rocks of our planet, we see a strong bifurcation with the production of a mass-independent sulfur isotope signature only in rocks that are before the Great Oxidation Event, in the Archean rocks of our planet. And then, of course, that signature disappears after we have an oxidized atmosphere, and we end up with signatures within the rocks of our planet that largely are at zero. There's no longer any excursion in the Cap33 delta signature, as we see in Archean rocks. Kidd Creek, of course, a famous Archean massive sulfide deposit, has been studied and studied and studied again in terms of understanding not only its delta sulfur isotope signature, but this mass-independent sulfur isotopes. And as you can imagine, being a deposit from 2.7 billion years, it's no surprise at all, and has long been known, that the rocks of Kidd Creek have a non-zero Cap delta 33 signature, as they should. Shown here for some of the pyrite and massive pyrite from Kidd Creek, we see that characteristic Cap33 excursion that indeed identifies these as a Archean sulfur source. What was interesting, so no surprises there in the rocks, what was interesting and the really creative insight of Long Lee is he wanted to look at the fluids as well. Take a look at those macroscopic fluids discharging in the present day. And when he did that, we'll just show you that here. Again, I'm showing you the rocks here. When Long did the waters, which we're showing you here in blue, this is where we got a real surprise. This was the first time anyone had ever identified a mass-independent sulfur isotope signature in water. And as you can see, the water of Kidd Creek tracked exactly the Cap33 signature of the rocks. It's different in the Delta 33 and Delta 34 signatures, and I'll show you that later if you're interested in the details of that. That's actually a function of the microbiology in these systems. But what I'm focusing on right now is just the y-axis and the fact that the waters track the Cap 33 signature coming from the wall rock, telling us that in this very hydrogeologically isolated system, with mean residence times on the order of billions of years, the sole sulfur source for the waters in this system appeared to be derived from the rocks themselves. And in fact, this came back to the issue of radiolysis. As radiolysis, as we talked about earlier, is taking place in these systems and producing all of the hydrogen that I've told you about, of course, by mass balance, it's producing a lot of these reactive oxides as well. And indeed, it's been shown in experimental literature that as you get that uranium, thorium, potassium decay, producing alpha, beta, and gamma particles, breaking apart the hydrogen molecules to produce, the water molecules to produce hydrogen, the oxides as well are very reactive. They hack the sulfide minerals as well and provide a mechanism by which that sulfide minerals are then oxidized to produce the dissolved sulfate that we find in those fluids. And so in a fascinating study then in 2006, what Long really showed is that radiolysis then provided a closed loop for this system, producing not only the hydrogen, but also producing the attack on those sulfide minerals, and thereby radiolysis providing within the system at Kidd Creek a coherent and long-term mechanism capable of supplying both the electron donor and the electron acceptor for life within the system. And it really allowed us, as you'll see in this bullet here outlined in white, to begin to focus on radiolysis, not just as a driver of deep subsurface hydrogen cycle, but also for the first time as a driver of the sulfur system within these as well. What I want to show you in the last few minutes today is that we're now extending our understanding of these kinds of radiolytic processes to the carbon cycle within a system like Kidd Creek as well, in a paper that just has been published, I think I sent to you beforehand with the advanced materials for this lecture. The carbon cycle was actually where much of this work started. In fact, back in some of the papers that came out of my PhD, Kidd Creek was one of the first continental systems in which we were able to show abiotic organic synthesis, not only for methane, but also for some of the lower alkanes as well, for ethane, propane, and butane within these systems. More recently, however, in 3 papers that were published just in 2021, we've been able to extend our investigation of the carbon cycle within these systems, once again taking advantage of this deep observatory, which the mine has allowed us to put in place at 2.4 kilometers below surface at Kidd Creek. And in particular, what I'm going to talk about now is indeed that most recent paper, which gives us an additional window into the abiotic carbon cycle. This is work that we've done with Verena Haar and Kai Heinrichs at Bremen, and particularly focusing on acetate and formate within these fluids. When people think about these deep systems, sometimes people assume that they are carbon poor. In fact, they're anything but. There's extensive sources of carbon within these systems. There are graphitic horizons, there are carbonate fracture minerals, there's even low concentrations of dissolved inorganic carbon on the order of 30 to 60 micromoles. The question is not whether or not carbon is present. The question is whether or not carbon is present in a form that can be of use to microbiology and understanding the ultimate sources and sinks of these carbons, and whether or not they are indeed used by microbiology. As we began to investigate not just the gases, the methane, ethane, propane in these systems, but the dissolved organic carbon, what was fascinating about this system early on is to discover that we had a very unusual and a very simple organic soup, as it were. Essentially, the dissolved organic carbon, the two major compounds that make up almost all of the dissolved organic carbon at these systems are shown here. The DOC is in orange and the acetate and formate together in blue. DOC is up to about 2 to 5 millimolar, but 50 to almost 90% of that is just acetate plus a little bit of formate. As I say, a very simple organic soup. The concentrations of acetate are actually extremely high, 1 to 2 millimolar, 1200, 1900 micromolar. Concentrations of acetate high as well, but the two of them together really accounting for the bulk of the organic carbon load. Very interesting characteristics compared to acetate and formate found in other systems as well. If we compare them for a moment to sedimentary basin brines, they're quite different. Sedimentary basin brines typically have very high levels of formate. Ours have very high levels of formate. The sedimentary basin brines rarely have high levels of formate. In addition, the Kidd Creek system has none of the other suite of organic acids typically found in sedimentary basin brines. We have no detectable propionate or butyrate or any of the others. The Kidd Creek systems differ from the hydrothermal vents as well, in the sense that hydrothermal vents typically have a higher ratio of formate to acetate. Typically something as work by Jim McDermott and Susan Lang has shown formate on the order of tens to hundreds of micromolars and much lower acetate levels. In contrast, these systems have very high levels of acetate and much smaller levels of formate. In fact, typically a 2-to-1 ratio for those two components. Indeed, as we've mentioned too, we've been monitoring these for over 10 years. And the other extraordinary thing is that this kind of picture that I'll show you here, which shows this compositional and isotopic mass balance, showing that the acetate and formate really control the dissolved organic matter within these systems. There's largely been no change in those concentrations and no change in those isotopic signatures. They're absolutely robust year after year after year after year. This in and of itself suggesting perhaps that these are not compounds that are being utilized in a very active microbiological sense, or I doubt we'd see that kind of continuity. More important, however, in terms of trying to understand the origin of these fluids and what's controlling their compositional and isotopic composition are these diagrams. These show us that the acetate in the Kidd Creek water is not only a very high concentration, but some of the most enriched isotopic signatures for acetate that have ever been identified. Very, very, or highly enriched, very weakly negative, if I might put it, delta C13 signatures accompany these very high concentrations. So at Kidd Creek, both the high concentrations of acetate and formate, the predominance of acetate over formate at a 2-to-1 ratio, and these highly C13 enriched values for acetate are all unprecedented in the literature. There's no other setting in which we've seen such a phenomenon. In terms of even comparing them to other subsurface sites that have been accessed by mines around the world, there too, they're quite unusual. At other sites where we've taken a look at the isotopic composition of acetate, for instance, either within, this is another site in ultramafic rocks on the Canadian Shield called Thompson Mine in Manitoba, or if I compare these to data that we have produced out of the work in our gold mines in South Africa, there we see the delta C13 values typically overlap with those of the macroscopic delta C13, the graphitic delta C13 in the rocks. But again, not so at Kidd Creek. We have delta C13 signatures here that are very different from what's found in the rocks as well. So really an unusual and, as I put, a rather unprecedented set of characteristics for the acetate and the formate in these systems. In the paper, you'll see we tried to be very comprehensive to try to understand the nature of these compounds in the context of potential microbiology, thermogenic processes, all kinds of processes. And all of that's dealt with exhaustively in the paper. But what I want to do here is to address just the one aspect of the paper that really was, I think, quite exciting and a new angle on these. And that was brought to our attention by our colleague, Benedicte Menez. She had spotted in the physical chemistry literature a fascinating set of experimental studies. It turns out that the physical chemists were doing more than we were as geochemists and biogeochemists. We've been looking for a long time at radiolytic production of hydrogen, but they've gone further than that and taken a look, done experiments looking at radiolysis in the context not just of producing hydrogen, but also of producing some of these other organic compounds. And more specifically, this particular paper, it's one of a series that this group has done, had took a look at the production of acetate and formate and been able to show that radiolysis in the presence of carbonates, in particular calcium carbonate, produces not just hydrogen and not just sulfate, but also a number of organic anions, and specifically acetate and formate. And so, although this is early stages yet, these radiolytic mechanisms are yet not well understood. The production as a function of different rates of alpha, beta, and gamma radiation have still to be elucidated. And certainly no one yet has jumped in and done what I'm sure will be some very fruitful research trying to understand the stable isotope fractionation factors associated with these reactions. But nonetheless, what we were able to do in our paper was to at least do a first order math balance. For many of these sites, we have a good handle on the radiolytic hydrogen production rate. And if we do assume for a moment then that radiolysis is driving not only hydrogen and the production of sulfate, but also through to the abundant calcium carbonate fracture minerals in these systems, potentially producing acetate and formate as well, if we take those rates of hydrogen production, couple them with the production rates found in the Costiaglia paper, we can come up with a first order estimate of how much acetate and formate might be produced in our systems. And what's interesting, again, the details are in the paper, but if we start first in the Witwatersrand Basin, there, I think I mentioned, we have fluids residence times on the order of typically tens of millions of years. If we take that time scale and the rates of acetate, formate, and hydrogen production, then we would predict production of those compounds on the order of hundreds of micromolar, which is in fact what we find in Witwatersrand Basin. In contrast, as I mentioned, as we go to Kidd Creek, we see concentrations of acetate and formate that are an order of magnitude larger, on the order of 1 to 2 to 3 millimolar concentrations of acetate and formate. And indeed, if we do this first order mass balance using the kinds of much older residence times that we found on Kidd Creek, residence times on order of hundreds to even billions of years, that could account for these higher levels of acetate and formate that we find there. So at least on the first order, it would imply that these radiolytic reactions might be a very, very fruitful way of trying to understand some of the abiotic carbon compounds that we find within these systems that we're not capable of explaining from all of our standard interpretations from biogeochemical processes. So it's essentially shining a light on a very unusual system in the deep crust. These very ancient, very saline Kidd Creek systems that have been suggested to be places where habitability can be sustained by deep subsurface hydrogen and sulfur cycles driven by radiolysis. This most recent study is showing us that it may go further yet, and radiolysis may also provide us with mechanisms to sustain a deep carbon cycle. And in particular then, of interest to those of us who are trying to understand ways in which deep subsurface microbes might in fact be sustained on geologic timescales deep in the subsurface once they've been cut off from the surface photosphere. Or further yet, provide us with models of planetary habitability on other planets and moons, where perhaps we might have the development of a chemosynthetic life, but where photosynthesis may not have arisen. So particularly in planetary sciences and astrobiology right now, as we are planning investigation of things like the plumes of Enceladus and understanding the habitability of some of the moons of Saturn and Jupiter, there's a lot of interest in understanding not only what life might have looked like if it was like Earth, photosynthetically based, but what life might look like if it in fact arose on a planet where there was no opportunity for photosynthesis, but where chemosynthetic reactions of water-rock reaction might control habitability, where we may have life not so much as we know it on Earth. And so to conclude then, in the 2 minutes remaining to me, it's been a fascinating series of investigations over the years. The last 30 years we've been working in these systems. I tried to give you a very quick overview today that I hope might be of interest to the wide breadth and diversity of audience interest here today. But certainly what we're seeing in these deep systems is that they're providing us with an opportunity to take a look at preservation of fluids and noble gas components on much larger and more ancient timescales than it's available to us from looking at the marine crust, which is necessarily, of course, no more than a couple of 100 million years old. It's providing us with novel samples of very ancient systems, but I focused more today on this context of habitability and the link to the understanding of subsurface life, and in particular the way investigation of processes of water-rock reaction in these systems provides us with an understanding of the types of electron donors and electron acceptors from water-rock reaction that might sustain subsurface chemolithotrophic life. And then in particular then, in the context of astrobiology, these kinds of systems provide us with analog environments that allow us to really begin to understand how life might be not quite as we know it when we begin to take a look at other planets and moons in our solar system. And so I'd like to leave you there. I mean, the astrobiology context of these is quite a fascinating part of this right now. As you know, next, we have just had one of the 3 Mars missions go into orbit successfully around Mars about 2 days ago. We have 2 more missions coming, including the landing of the Perseverance rover next week on the 18th. And so a lot of these Earth analogs are providing us with a fascinating way to try to test out some of the process-driven issues that may be the answer to searching for life on other planets and moons in our solar system. I'd like to thank a large group of people. As you can imagine, over 30 years, the various postdocs and contributors that have contributed to the work that you saw here today are many. I've named a couple of them as we went along, but I want to as well thank a recent program that we're running within Canada through the CFAR exploration program, and that's called Earth2Earth. It's an international program where you have people from all around the world that are participating in these questions of subsurface science and exploration, ranging from understanding water, rocks, life, and energy, both on our planet and further out in the universe. It's co-directed by myself and Dr. Jack Mustard at Brown, but as you can see, there's a large number of international collaborators here. This particular picture is taken of them all about 2 kilometers below surface at the Sudbury Neutrino Lab in the Canadian Shield. And so with thanks to that group, I look forward to taking any questions that may come up. Thank you so much for this opportunity. It's really been a wonderful chance to reach out during these times of COVID.
Thanks so much, Barbara. Really fantastic talk and terribly exciting set of observations. I have a whole load of questions of my own. There's also some questions in the Q&A. So let's jump straight to those. Maybe we could start with like a sort of basic one in some respects, which is to ask, how is the volume of the subsurface groundwater been estimated? Maybe related to that very first kind of schematic you showed. And could it be much larger even than the current estimates imply?
Absolutely. I mean, it's one of the things that we emphasize in this Earth2Earth program, that one of the fascinating things is, to some extent, there are aspects of our subsurface that we have almost no coherent understanding on. If you take a look at the number of actual measurements of porosity and permeability. First and foremost, they're patchy around the world. And then when you go below 1 kilometer depth, few and far between. So there's a large gap in our understanding of the actual data-driven issues of porosity and permeability. So if you're asking about the Gleason et al. Paper, what they did was to take tritium measurements and then use depth distributions for porosity down to 2 kilometers. Many of these exist in the world. They're dependent upon the assumption largely that you're dealing with a continuous porous media. And so they're very applicable, for instance, to sedimentary basins. But it's arguable whether you can take a model that's developed based on a porous media in a sedimentary basin, which is largely, as I mentioned, based on the idea that the water will be distributed in the pore spaces between the grains, and then extrapolate that into a fractured rock system to great depth. And largely, really, we can't. And so typically what's been done instead is to, as you go deeper into fracture system, makes those estimates based on global porosity estimates for rock. These two are really far and few between. They're good. The studies that exist are good, but we really do have a paucity of data. And so, in fact, one of the key pillars of this group that I'm highlighting here is really to try to attack that question, to think about what do we really know about the subsurface architecture? More importantly, what don't we know? What are the key gaps that need to be filled? And then how can we begin to advance our estimates of groundwater in the subsurface beyond these really very back of the envelope models that exist at the present time? So I think it's a real pointing, hopefully shining a light on something that very often we think we know more about than we do. If you scratch this, you'll find there's a real paucity of data, and to turn that around into a positive sense, then a real opportunity for some exciting and novel research. What could be more important to our planet than understanding the nature and distribution of where we have water? But it's still a real frontier.
Fantastic, thank you. And sort of maybe following on from that theme of how much there is, there's a question asking essentially where's it come from? So what's the provenance of these ancient deep crustal fluids, and were they ever participating in the surface hydrosphere?
Well, we know for sure from the ground, from all of the work that we've done now, that they have been cut off from the surface photosphere on very, very long time scales. There's no tritium or anything like that in them. And the noble gases, of course, provide us with a really precise tool. We would immediately see if there was a surface connection in the present time. So we know that whatever those waters are, they are not young. That said, even I was surprised by the really profound ancient nature of what we found. Essentially, like any water, the residence times that are given are an average. So they represent the mean residence time of the bulk fluid. So in some ways, that only provides one of three really important things you'd want to know. It gives you the residence time of the bulk fluid, but it doesn't tell you either the oldest drop or the youngest drop. So really, if you think about it, the three components we'd really want to know to understand the system. Now at Kidd Creek, we actually have some fascinating information on what might be the oldest drop. And as I referred to it there, in particular, the neon isotopic signature and indeed the xenon isotopic signatures are actually comparable to isotopic signatures we found in primary fluid inclusions. Primary fluid inclusions that represent, in some cases, 2 or 2.6 billion year old metamorphic fluids in those rocks. And so for Kidd Creek, it's one of the few systems where we actually do feel we have a good angle on the oldest drop. And the oldest drop then would likely represent the most conservative components, these noble gas signatures that show us we actually have some components that are as old as the rock itself, or at least as old as the last regional metamorphic fluids that went through those rocks. The bulk water trends younger, if you can call something that's 1.2 to 1.6 billion years old younger, but it does mean that the rest of the fluid represents the long-term water rock reactions that have taken place between the rocks and the fluid. The very youngest drop, This is something that we have yet to been able to constrain, largely because we really don't have good tools for constraining groundwaters on the order of hundreds of millions of years, but we're continuing to work and try to advance that. So it's a really good question. The important thing to understand is any groundwater is always a mixture. And so what we really want to understand are those three components, and that's the kind of thing that will provide us with a really important range of constraints on the system. In the context of life, for sure, it's always a mistake to assume that the microbes that you find are as old as the rock itself, or even as old as the bulk water. Really important to try to understand the youngest drop, because arguably, of course, that could have been what brought the life into the system. And so these need to be considered as separate questions in those full contexts to really understand the nature of life.
Great, thank you. A popular question is the presence of H2, I think you showed us that video of bubbling water early on, is the presence of that H2 problematic from the perspective of there being a deep biosphere, which might otherwise have utilized that available energy source and consumed it?
Right. We actually know quite a bit about this at this point in time. We do know, for instance, from South Africa, that it is entirely possible to have millimolar concentrations of hydrogen and yet absolute evidence of active microbiology. You'll see this, for instance, in the two papers I referenced from the deep South African gold mines, Lin et al. 2006, Chivian et al. 2008. Those were done with our colleagues, Esteban Herden at the University of Free State and T.C. Onstadt of Princeton, Tom Keefe of New Mexico. What it tells us is that in these systems, the rate limiting step for life is not the presence of the hydrogen donor. We see some sites where they've actually gobbled up all the hydrogen, but there we see actually differences in the nature of the microbiology. Where we see these most low biomass, cell counts on the order of 10 to the 2, 10 to the 3 cells per mil, low biodiversity, there we often see the presence of active microbiology in the presence of still high concentrations of hydrogen. So showing us that there's something else that is producing the brake for life. It may be, in fact, the presence of available carbon sources. We don't know for sure. It's one of the key questions in these very deep subsurface communities. What is actually the limits to life within those deep systems? In some of the ones, as I mentioned, between 1 and 1.5 kilometers, we see much more biodiversity, much higher cell counts, a much wider variety of microbial metabolisms, and with those systems, typically they have driven the hydrogen down below detection.
Great, thank you. As a question from Paul Rimmer, is there any way to estimate the fraction of reduced hydrocarbons in Kidd Creek that are biogenic versus abiotic in origin? And if abiotic is the source or production mechanism known?
Yeah, I mean, that's something I didn't talk about that here because that's been developed for quite a few years. Essentially, the closest plausible mechanism for producing the abiotic methane, ethane, propane, etc., is they seem to be consistent with Sabatier reaction producing the methane and then polymerization reactions such as Fischer-Tropsch synthesis responsible for the ethane, propane, butane, etc. That allows us to identify that in these systems, we have predominantly at Kidd Creek an abiotic methane mechanism, but that's not exclusively. We do see some production of biological methane, and typically the ways in which we identify that microbial methane and abiotic methane is by both temporal investigation. We actually see in these sites, many of them start out with a very abiotic signature, and then over time, there seems to be a bloom of life, and we see migration of those signatures over towards more microbial methane contributions. And relevant to the last question, as that happens, we see the hydrogen drop. So it's by monitoring those temporal and spatial changes that we're able to begin to get a handle on the relative contribution of abiotic versus biological methane at these sites. They're almost always a mixture. Kidd Creek is fascinating in that it's the one that has the highest abiotic to biotic ratio, but we go all the way over to other sites where it's almost dominated by the microbiology. New techniques for doing that are found in the clumped methane isotopes. And one of those papers in the earlier slide, published at the same time as this acetate and formate paper, is in fact a temporal study of the clumped methane isotope changes for these same sets of boreholes. Again, that's by Oliver Warr, the research associate that I referred to earlier in our laboratory. And that's a new approach to trying to address exactly that question. So if you're interested in how clumped methane is helping us address that question, I certainly welcome you taking a look at that paper, and happy to answer any questions about that.
Perfect. There's a question from Eva Stucken. How much difference does pressure make in the subsurface chemical reactions, and particularly in the organic assemblage?
Unfortunately, that's one of the many, many parameters that's really under-investigated. And I had the great pleasure 2 years ago of chairing the National Academy's report on the search strategy for life in the universe. And this was one of the key recommendations, is that the work that we've done so far, we've tended to focus on one parameter. We look at the thermal limit for life, or we look at the hydrogen limit for life, or we look at the depth limit for life. But what we rarely do is put them all together and understand that, in fact, the limits for life are going to be the net result of this complex matrix of parameters. And so this is really one of the key questions that you've raised there. What are the pressure limits, and how do pressure limits match up with temperature limits and the geochemical limits? So I have to say, at the moment, we really don't have good answers for that, but it's one in which we hope to see much more work taking place in the future, particularly as people begin to try to address these experimentally as well. Take a look at this combination of temperature and pressure in chemistry.
Fantastic. Do you have time for maybe one or two more questions? I appreciate.
Yes, I'm totally fine. Yeah.
Okay, thank you. It'd be lovely just to kind of ask a couple more of these. They touch on a couple of subjects we've not come to yet. One of which is how decoupled is the subsurface biosphere from the surface biosphere? I.e., how many of the components involved in the subsurface biosphere, hydrogen, carbon, or sulfur, are ultimately derived from organic sediments and thereby photosynthesis?
Again, one of the key pillars of this program, we don't know. We know in some individual places. In other words, in some places on this planet, we can give you some sense of the relative degree to which we're actually looking at this in what we would rather poetically call the hidden hydrogeosphere, the part that's actually cut off from the photosphere. And then there are other places where we can absolutely show you that what we're finding is, in fact, sometimes I jokingly say, not so much subsurface life as surface life gone underground. Meaning, there are sites, particularly in South Africa. I encourage you to take a look at those studies, in particular some wonderful work done by our colleague Gaetan Bourgani. He's not only a wonderful scientist, but a terrific photographer. Some of the best pictures I've shown you, and you'll notice, are credited to Gaetan. But he's also a nematode specialist. So he has identified those sites in South Africa in which we actually see the presence of nematodes. Now, that's a clear indicator that we have life that has colonized the subsurface. And so we know a little bit about that question in that place, or this place, or another place. But understanding what fundamentally controls those boundaries of the deep biosphere, where we see the penetration of life that can still be connected to the surface on a hydrogeologic cycle, where we see systems that have been cut off on some sort of a time scale, but are still using carbon sources that are derived from the surface photosynthetic cycle. And then where we see systems that are truly an ultra deep biosphere, where they're having to draw whatever they can get from the rocks themselves. We don't, on a planetary scale, understand yet all of the processes that control where those various divisions of the deep biosphere take place. But that's something that's really driving our work and the work of many wonderful teams out there. It's something that I hope over the next 10 to 20 years we'll begin to understand for our planet. Because again, it'll be deeply important for our understanding then of how we would go about and what our chances would be for understanding those same processes on other planets.
Fantastic. And in fact, that leads us nicely to Tamsin's question in the Q&A there. which is, could you say more about your thoughts regarding what signatures of life we might pick up from afar? So atmospheric measurements of exoplanets as they improve, for example.
Ooh.
So what's kind of the global signature of deep biosphere?
Okay, I'm just going to joke for a minute and tell tales out of school for a second. As we did that National Academies report, it was taking place so there are two major National Academies taking place in 2017 and 2018. And one was the one for astrobiology and the search for life in the universe, and the other was simultaneously the astronomy and astrophysics one. And we had a really fun joint session together that we jokingly called, what do you mean, oxygen? And what we're joking about there is the fact that the immediate question is, of course, and it makes sense, scientific sense. Of course, if we find evidence for oxygen on an exoplanet, we're going to be excited. However, we need to fundamentally bear in mind that if that had been the strategy on looking for life on this little planet of ours, well, that would have been of no use whatsoever in the first 2.5 billion years of our existence. There was no major oxygen line in our atmosphere, and yet we had abundant life. And so in some ways that framed the very wonderful and productive conversation going on between our two communities. Of course, we're interested in life as we know it, but we have to also find ways to ensure that we invest in this search for life elsewhere, that we think very clearly about biosignatures that are more agnostic. And by that, we mean not weird life, not pivoting to only look for weird life. No, instead to try and make sure that we're looking for universal biosignatures, biosignatures that would allow us to identify life that might not only be photosynthetic as the dominant life on our planet is now, but might also ask the question, if you had life, for instance, that was driven only by water-rock reaction, only by chemosynthesis, what then would be the biosignatures that we would look for? And I think, again, that means it makes it one of the most exciting times, not only in the discovery of these exoplanets, but really in the communities coming together and having really powerful discussions about how we can learn from each other about ensuring we're looking in the most broad way possible so that we have the most chance of identifying not only life as we know it, but perhaps life as we belatedly discovered is here on this planet. But for instance, even when I was in high school, we weren't aware of this life. And that's a very humbling and I think important lesson for we scientists to take on board. Even on this Earth, there's still much to explore.
Fantastic. And I think that's a fantastic kind of note to end on as we look to the future of exoplanetary sciences and indeed Earth sciences into the deep biosphere. Thank you very much again, Barbara, for that really fantastic talk. Brilliant, exciting observations. I think we all look forward to seeing how the field moves forward. And thank you everyone for attending. We'll be running again in 2 weeks' time, February 25th, where Jibrain Labidi and James Dotton will be speaking about volatiles in Earth's mantle, primordial or subducted. So hopefully see you again then. Thank you again, Barbara, and take care, everyone.
Thank you so much. Much appreciated.